Hi everyone, and welcome to our review on carbohydrates. This is part 4 in our little carbohydrate series, and what we're going to be doing in this video is finishing off the polysaccharides. So you can see the 3 bits of our specification we're focusing on in this video: synthesis and breakdown of polysaccharide by forming and breaking those glycosidic bonds, structure of cellulose, and how the structures and properties of cellulose molecules relate to its function in living organisms. If we have a look at how we actually make this cellulose, then it's not quite as simple as all the beta glucose molecules just joined together in a long chain. There's a little quirk to it. And the quirk is that when we look at it, here's our first little beta glucose, and there's our second beta glucose. Now, what we find is, when we draw beta glucose, if you remember, we've said put the little oxygen in the top right and then draw the rest of your ring structure. So the beta glucose on the left of the diagram, absolutely fine. We see it, it's there, we know it's a beta glucose because the hydroxyl group is above the ring. If you look at the molecule on the right of that diagram, the oxygen is not in the top right position. The reason for that is that when cellulose forms, that second beta glucose molecule is actually flipped by 180 degrees. So if you look, the oxygen now sits in the bottom right corner. So all we've done is we've just flipped it by 180 degrees. What that means is when we then form our little glycosidic bond between, obviously, the 2 hydroxyl groups between the 2 beta glucose molecules, instead of having this spiral structure that we saw with our previous polysaccharides, we end up with a straight chain. So you can see here that with our little molecules at the bottom, so we've got our beta glucose with the oxygen at the top, beta glucose with the oxygen at the bottom, beta glucose, oxygen at the top, and that would continue along the chain. So because of that, we end up generating a straight chain because of how those glycosidic bonds now sit. So as those beta glucose molecules are rotated by 180 degrees in each instance, then we form these beta 1,4 glycosidic bonds, and that helps to prevent the chain from forming that spiral structure. So we get a straight chain rather than the spiral because of this. When we look at the fact we get these hydrogen bonds still forming between those glucose molecules in the chain, that just provides additional strength to the cellulose structure. We can also get hydrogen bonds forming between different beta glucose molecules in separate chains, and that's going to build in yet more strength. So what we actually have is that hydroxyl group that sticks out from carbon 2, and that's then going to enable that hydrogen bonding to different cellulose chains. So what we can see there is we're getting hydrogen bonds in 2 regions: within the actual chain itself between the individual beta glucose molecules, and hydrogen bonds that form between different beta glucose molecules in different chains. Both of those are going to provide strength to the overall structure. In terms of why cellulose has evolved to be the chemical that's used to form these plant cell walls, then we have a few key things to remember here. Firstly, those micro and macro fibrils that make up the cellulose have a high tensile strength. The reason that they've got that high tensile strength, they've got the glycosidic bonds, obviously between the individual beta-glucose molecules, and hydrogen bonds, both within and between those different chains. Because they've got that high tensile strength, that's going to help those cells with cellulose in the walls to avoid bursting. And remember, that's going to be really important because we're going to have water moving into our plant cells, making them turgid. The last thing you want to happen is for that cell to burst when they are turgid. So the cellulose helps to prevent that because of the high tensile strength. Secondly, it's insoluble and it's unreactive. So that means that we can have this structure to make up the cell walls that won't be dissolved by any liquids in the environment around it. Quite important. And it is flexible. Now, being flexible does mean that there's that little bit of movement that is necessary for obviously the plants to be able to move in the breeze without just snapping outright, for example. In terms of why our plant cell walls have to be strong, the plants themselves have no skeleton. The only way that plants are able to grow vertically is because they have this pressure created by the vacuole pressing out on those cell walls, and the cell walls basically resist bursting. So that enables the plant to be supported. Our cell walls are also going to be permeable, which means water and mineral ions are able to move into those cells. If they weren't able to allow water and mineral ions to cross the cell wall, we would have a situation where the inside of the cell was cut off from everything it needed to carry out all those chemical reactions to keep it alive. So obviously having that permeability is going to be very important for general life of a cell. Another thing that could happen in terms of our plant cell walls is that we can actually reinforce the structure further through the addition of certain other substances. Some of these you will have already heard about lower down the school. So for example, we have lignin. Now lignin is one of those chemicals I'm expecting you heard about at GCSE with regards to xylem vessels. Xylem vessels are the ones that transport water, and by laying lignin along those cell walls, what we do is we waterproof the xylem vessels. So lignin waterproofs the xylem vessels. It's got a second function as well, in the fact that when the lignin is laid down, it thickens those parts of the plant. So we apply lignin to regions like the woody trunks, for example, of trees, and that just provides additional strength because trees are quite big and therefore quite a great amount of mass is weighing down on that one trunk. So by using lignin in those cell walls, it gives it that additional strength to be able to withstand the pressure from obviously that increased mass above. We also have these other two chemicals that you probably haven't heard about, Chitin and suberin. Now, both of these are waterproofing agents, and the way that they waterproof the cell wall is by blocking the spaces in it. So ordinarily, we've got these little spaces that allow substances to cross the cell wall and therefore obviously makes the cell wall permeable. If we apply Chitin and suberin, then it will block those spaces and therefore make that cell wall waterproof. The final bit we're going to look at are exoskeletons. Now, exoskeletons are structures that you find on insects and crustaceans, and exo just means external. So exoskeletons are where they've got their skeleton basically on the outside of their body. We have an endoskeleton, it's on the inside, we have skin over the surface, etc. Whereas insects and crustaceans, their skeleton is on the outside, hence exoskeleton. And these exoskeletons are made from a substance called chitin. In terms of the actual structure of chitin, then it's got this thing called an acetyl amino group. So you can see the chemical formula for it there, you don't need to memorize that. So we've got this acetyl amino group in place of the hydroxyl group that we would normally find on carbon 2. So what we have there is this very subtle change. We've just swapped out hydroxyl group for this acetyl amino group, but we've now made an entirely new chemical structure. So what we end up with are these long chains that run parallel to one another, and we then have cross links forming between them. So quite similar to cellulose in some ways, but we get these acetyl glucosamines that are going to form the cross links between the parallel chains there.